Good afternoon. Welcome to the lockdown series lecture or the panel discussion that we are having on behalf of the Neuroscience Center, the National Council of Science Museum, Ministry of Culture, Government of India. It gives me immense pleasure to welcome our distinguished speakers, scientists from the Institute of Genomics and Integrated Biology, a CSIR lab from Delhi. My colleague Juma will be introducing each of the speakers. We are in hopefully we are coming to an end of the pandemic. Some kind of a light at the end of the tunnel is appearing to be seen. So on this topic, the COVID and what the Sero Survey, the vaccine development and awareness, and diagnostics. These are the topics in which three of our distinguished scientists will be speaking. One of the reasons why we took up this topic is there's an accentuated curiosity regarding people are waiting for the vaccine, and there's lots of so-called infodemic which is getting spread across the media. So let's have some panel, some experts who will be able to communicate the exacting science that is actually required to be communicated, rather than the WhatsApp message, huge amount of WhatsApp message that comes in. Quite a lot of them is fine. Perhaps 10 times that is perhaps is found something wanting. So that's the reason we are doing this. Interestingly enough, India always excels when it comes to some challenging times. It's happened during our, there was post the nuclear program, for example, when we faced the what you call as the emergency cuts from US and other things, we developed our own supercomputing and things like that. But when we went into the lockdown somewhere in March, our numbers for testing was so very pathetically low. But then, the first thing what happened is one young lady, Dr. I think Meenal Bhosle, way back in March, she started something just before she discovered a kit and then went into for delivering her own baby. There's a major kit that was developed by Institute of Genomics and Integrated Biology using the CRISPR-Cas technology, which incidentally has won the Nobel Prize this year. So I think I'm sure the speakers are going to be speaking on that. They're going to be speaking on what the three vaccines which are likely to be introduced sooner than later in India, and also of the kind of a Sero survey that has been done extensively both by IGIP and several other institutes. What the Sero survey means, what is that so-called, what we call as a herd immunity, which everybody is looking up to. These are the issues which our speakers will be speaking. Before I hand over to my speakers, I would request my colleague Juma, who has managed to put this across, to introduce our speaker. Let me also tell you that the Neuroscience Center is a constituent unit of the National Council of Science Museum. We were a part of CSIR, in which IGIB is at one point of time, but then subsequently it was decided that science communication aspect of it and the science museums and science center should have their own autonomous body. That is how the NCSM has been formed, but that umbilical connect it's still there between CSIR and NCSM. So I hand it over to Juma now. Juma, please.
Thank you. We have Mr. Manas Bagchi, Curator and Project Coordinator of RSC Calicut as the organizer from our side, and Dr. Souvik Maiti, Senior Principal Scientist in Nucleic Acid Biophysics from IGIB. And we have three speakers. First, Dr. Shantanu Sengupta, who is now the Senior Principal Scientist in Cardiovascular Disease Biology. He is interested in identifying pre-diagnostic markers for cardiovascular disease using genetic, epigenetic approaches. Today, he will be discussing on the importance of serology to assess the burden of COVID-2 infection, and will touch on the Pan-India Sero Survey that they have just completed. Our next speaker is Dr. Vinod Skaria. He is the Senior Scientist in Genome Informatics. He is a clinician and computational biologist. His research interest includes understanding the function organization and regulation of genome and how the genomic variations could potentially impact them. He is also involved in creating novel methods and resources for analysis and annotation of genomes and understanding the functional impact of those genomic variations. Today he will be discussing the insights from genomics of SARS-CoV-2 for genetic epidemiology in India, and would be touching upon the utility in diagnostics and vaccines going forward. And our third speaker is Dr. Jyoti Chakraborty. He is senior scientist in genome editing, stem cells and organoid biology. He works on the development of efficient tools for genome editing, the systematic dissection of factors that govern the embryonic stem cell fate, and the use of this organoid to address the developmental questions. Today, his topic of discussion would be repurposing the genome editing methods of SARS-CoV-2 diagnosis. Then he will give a brief outline about their work that's going on the paper strip based COVID-2 detection using the CRISPR. Now, let's welcome the speaker.
Dr. Baiti, I think we can now.
Hello, good afternoon to everybody. And I would like to start with thanking Mr. Bakchi, actually, as well as Mr. Kenneth, to give CSR IGIV outreach program to start or to have a collaboration to take our science to, most important community of our country, the school children, which basically we miss when we are in the laboratory. So this is a good opportunity for us also, and I hope our colleagues, our friends will be able to address all the curiosities, whatever they have. I'm extremely happy to part of this program. I will hand over to other speaker. Thank you very much.
Dr. Shantanu or any one of you, one of the three of you.
Deva, can you start? We can have diagnostics first, and then we can.
I think better is Vinod if you can start. I was just preparing mentally for that. Kind of.
Yeah, I think perhaps people are also waiting for the vaccine thing, so I think will be the right person to.
Sure, I'll go ahead. So, thank you, Neuroscience Center, for this opportunity. And for the next 10 to 15 minutes, I will sort of discuss about the genomics of SARS-CoV-2 in India and what we really understood from it. I hope you can see the slides.
Yes, please. Please go ahead. Yeah, we can see.
Yeah. So, what is really remarkable in this pandemic compared to other epidemics that has occurred across years in the last decade or so has been that it is one of the pandemics where there's a paradigm shift in understanding the pathogens. And this paradigm shift is essentially because of the sort of merging of two sciences. One is microbiology, which is an age-old science, and a new technology or a new science, which is genomics. And to give a perspective of what really is the paradigm shift in here, I will sort of exemplify this point that traditionally the investigation of all pandemics, or even for that matter, even any outbreak, has been through traditional microbiology, which includes culture, isolation, or purification of microorganisms before the identification happened. And what really changed in this pandemic is that there was nowhere in the early pandemic attempts to even culture or isolate or purify this organism, but rather directly make the sequences available, and the sequences which essentially what identified the pathogen. Now, this has been made possible because of one, the attempts to increase throughputs of sequencing, where we can sequence probably terabytes of sequences in less than 24 hours. And of course, methods to computationally assemble this data into organismal genomes, which can now enable people to understand it better. So now, to give a brief timeline of what really happened in the early pandemic is that the patient sample, which is a bronchoalveolar lavage, was essentially directly taken up for RNA extraction and then to sequencing and then further to analysis. Now, this is what really paved way to enabling the sequence of SARS-CoV-2 available very early in the pandemic. And this essentially is something that is very remarkable, because in probably weeks' time, the data for the Wuhan Q1, which is the initial SARS genome, was available. And of course, the paper on this attempt and this genome annotation came much later, back in February. And that's essentially how we know that what is the identity of this pathogen and how this pathogen is different or similar, for that matter, with other coronaviruses which has been discovered in sequence before. Now, the other remarkable thing that has happened is that it is not just one genome that happened, but genomes that have been continuously sequenced across different time points and across different regions of the world. And one thing that has been consistently made from these genome sequences was that the virus mutates, and the virus mutates as a factor of evolution for all organisms. And this mutation rate or substitution rate occurs at a quite sort of uniform fashion across the genome. And this is just an example of a few genomes from early in the pandemic, which sort of suggest how these mutations have occurred in different parts of the genome as they moved forward in time and space. Now, what is really that need to be understood is that if you think the virus can mutate, and the mutation is essentially an evolutionary principle that all organisms follow, the only difference is the rate at which each organism mutates. Now, of course, being an RNA virus, it has a quite constant rate of mutation, and this mutation rate is quite same across the estimates from different parts of across the world, and it's approximately around 3.7 or approximately 4 per month, or in other words, approximately 1 every week. So you could imagine, as the virus gets transmitted from individual to individual over a period of time, it accumulates mutations, and at some point of time, some individuals spread it much further, or in other words, it's a super spreader event which spread to a large number of people, and that's essentially how a genetic lineage emerges. Now, if you look at the vertical points in time, which are essentially points in time, right? If the virus was transmitting at a constant rate, you would have an equal proportions of all of these genetic lineages, because the rate of transmission is constant and the rate of mutation is a constant. Then the distribution of each of these genetic lineages at any point of time should have been constant. But that's not what we see. And what we see is essentially a skewed distribution, as you see here in the second vertical panel out here, which essentially suggests that there have been some points where there have been super spreader events or emergence events which give rise to a particular genetic lineage. And this is essentially what is really important, because given the constant rate of mutation and given the constant time of transmission, you can sort of predict not just where did this emergence event occur, but also be able to track both in time and space how and where did this happen. Now, people have done this remarkably over the course of the pandemic, and this is essentially from the early pandemic up till around April, from over 2,000 odd genomes which are available from across the world from many different countries. And what you see here is largely the genetic lineages at that point in time could be distributed into 10 genetic lineages, as you see here, marked with different colors. But of course, some of the genetic lineages were disproportionately larger than the other lineage. The big blue blob that you see in the top panel of this chart is essentially a genetic lineage, what we call as a A2A, which essentially had a European sort of ancestry. Now, this is useful because early in the epidemic, each of these genetic lineages did have some kind of a geographical preponderance, with the A group mostly from the eastern part of the world, the B group mostly from China and Southeast Asia, and so on and so forth. So therefore, early in the epidemic, this is also useful to understand from where did this virus come into a country, or what we call as introductions into a particular population. But of course, as we move later, many of these events would change, and therefore you could look at the dynamics of each of these genetic lineages and also make useful assessments about the progression of the disease or the pandemic across in time and space. Now, of course, by doing genomics, you can, of course, understand the evolution of the pathogen. You can understand the origin of infection, both in time and space, and also able to understand emergence events or outbreak events, apart from a lot of other things like evaluating diagnostics or even rational design of diagnostics, and of course, looking at vaccines and therapies. I'll briefly touch upon these points later in my slide. Now, of course, to do sequencing, you need to do sequencing at high throughput, and that's what has been made possible, this revolution in genomics for SARS-CoV-2. Today, we have close to around 200,000 genome sequences of SARS-CoV-2 and almost doubling every other month. And one of these techniques that is also initially standardized in our lab has been this approach called as COVID-seq, which can sequence anything from 300 to 3,000 samples in 24 hours and at quite remarkable accuracy and cost. And when we come back to India, we have at this moment close to around 4,000 odd genomes sequenced and deposited from multiple different laboratories across the country, from multiple different states and across multiple different funding or organizational structures, including hospitals, research institutes, individual laboratories, so on and so forth. And what you see from India is not really 10 different lineages. 10 different lineages are found across the world. In India, we find only 6 out of these 10 genetic lineages. And apart from that, we have a unique genetic lineage marked here in violet, which we call as the I or the A3I clade. Now, as you see here in a factor of time, the I or the A3I emerged in India sometime in early March or late February, and sort of became a predominant genetic lineage, at least in some states, as you see here. But nevertheless, even today, the largest genetic lineage in terms of the numbers is the A2A genetic lineage, which essentially traces its ancestry from Europe. So, in summary, India has a quite unique or distinct genetic lineage, which is called the IA3I. Now, we could really trace back IA3I lineage from India across different periods in time and across different states, thanks to sequences which are made available by different laboratories in each of the states. And what we find is quite remarkable difference in the predisposition or the preponderance of these particular genetic lineage in different parts of India. Now, this is from early April, May, where you see a large proportion of genomes from Telangana, Delhi, and some ways Tamil Nadu and Karnataka belong to this particular genetic lineage, which is quite distinctly unique to India. When I say distinctly unique to India, it doesn't really mean that it emerged or it was first found in India. It is also found in many other Southeast Asian countries and, of course, some countries in the Asia Pacific, including Australia. But nevertheless, in none of these countries it has become a quite distinct and large lineage. What we also could track in time and space is that the emergence of IA3I occurred from a superspreader event late in February, early March, where it has spread to multiple different states. As you see here in the right panel, there are multiple different states colored in different colors, but nevertheless, all tracing its origin from almost a single point event of emergence. So in other words, we know this has happened through a superspreader event. What we could also do using the genome sequence, as I said, we could trace lineages and genetic mutations or mutational fingerprints. You could now trace the transmission of the virus from across country as a factor of space and time. In other words, you can trace how from each of these cities, the virus has actually moved with people who are infected to another city and so on and so forth, and how did the dynamics change over time. And the other question, of course, you could ask is how is the virus evolving? And I sort of stated that the virus evolves at a sort of constant rate of around 3.7 per month. And each of these genetic lineages has subtle differences in the rate of evolution or rate of substitution. And what we show is that IA3I had a quite significantly lower rate of substitution compared to the predominantly prevalent A2A clade in India. And what we sort of understood from this low prevalence was that eventually the IA3I would have a fitness disadvantage because it evolves much slower and then therefore would die out eventually. And that's what we have seen over time for this particular plate-agentic lineage. And of course, there are multiple other applications for looking at genome sequencing. And one of the important applications of looking at genome sequencing is to be able to understand the efficacy of diagnostics, and of course, in some cases, be able to rationally decide or design diagnostic reagents. Now, coming to diagnostics, real-time PCR is one of the mainstays for diagnosis of SARS-CoV2. And the principle of real-time PCR is essentially polymerase chain reaction, where you have a primer or a probe which can actually bind to the genome of SARS-CoV2, identify them, and amplify a small piece of nucleic acid, which is probed by a probe with a fluorescent label. Now, all of this is dependent upon the thermodynamics of the DNA-DNA hybrids, and then therefore genetic mutations in these hybrids could affect thermodynamics of binding or detection, and therefore the efficacy of the reagents. So what you could do is to essentially map all known primers and probes from genomes available across the world and ask this question: are there frequent genetic mutations in these sites which can bind to primers and probes? And using that, how can you sort of prioritize genetic reagents for testing? Or in other words, you could also use the same principle to go back and look at regions in the genome which are unlikely to have genetic mutations, and then therefore be able to design better reagents which are immune to genetic mutations at their sites. So this is what we have done, not just for the Indian genomes, but also for genomes across the world, and this was published quite recently. And before I close, what's really more important in the recent times is that people develop an immune mechanism to sort of contain the virus, and that's really important in sort of containing the pandemic moving forward. Either people could have immunity after an infection or immunity after getting a vaccine. But nevertheless, both of them are reliant on the two arms of the immune system. One is the humoral immune system, which is modulated by B cells, which make antibodies. And the other arm is essentially the T cells, which mediate the cell-mediated immunity. Now, coming to antibodies essentially recognize a particular region in the protein structure or protein sequence, what we call as epitopes, or what we call as, another word, antigens. Now, there could be two scenarios when we talk about antibody-mediated response. One is a scenario where an individual has enough neutralizing antibodies, and a scenario where you could have low or no neutralizing antibodies. In that particular case, an individual could be susceptible to an infection. In the first case, you could actually measure it through serosurveillance, and my next speaker will essentially delve deeper into serosurveillance. The other aspect is whether you could have a mutation in the antigens or the epitopes, which can actually change the recognition pattern for the identification by antibodies or neutralizing antibodies, and then therefore these viruses could then escape immune recognition, because antibodies recognize a particular motif or a particular region in three-dimensional space, and if you change the three-dimensional space, the antibody is not able to recognize that particular antigen anymore, and then therefore the vaccines could escape. Now, incidentally, there are a significant large number of antibody escape mutations now available in public domain. We have one of the largest curated list of such mutations across genomes. Now, one of the applications of this understanding in genomics is that you should be able to go back and look at very few cases of reinfections for SARS-CoV-2 in individuals. Now, reinfections essentially means that an individual had an infection, had contained the virus by his immune system, but then after quite some time, typically 3 to 6 months, he gets reinfected by another SARS-CoV-2 virus. Now, one of the first reports of SARS-CoV-2 reinfection in the country were actually reported by us, where we showed there was a significant difference in the genetic mutations between the two viruses in the two episodes. So this is for two individuals, two healthcare workers who had reinfections, and the panel essentially shows the points at which the genomes have changed for these two infections. And what's really more important is to now go back and understand what does these genetic mutations do in terms of immune escape. At least in one of these cases, we know one of the mutation is because of immune escape. It's a very well documented mutation. I think I'll stop there and hand over to the next speaker. But before that, to acknowledge a lot of people which made this possible. Thank you.
Thank you so much, Dr. Vinod. It was fascinating. I think now we'll shift to Dr. Shantanu. I'm sure he's going to be speaking on Sero Survey, which people are looking up to. Thank you.
I'm trying to
I think there's a present now screen. You can share.
I have done the present now, but there is it's not somehow showing up.
Can we do one thing? In that case, can we request Dr. Chakraborty?
Yeah.
Yeah, I can go ahead. Yeah.
Thanks, Dr. Chakraborty, please. Yeah.
So I hope you can see my screen.
Now, yeah, very clear. Now you can go for yeah.
And now can you see the slide?
Yeah, perfect.
Okay, good. So thank you very much for giving me this opportunity to speak today. I'll touch upon a little bit about our work in CRISPR Diagnostics for SARS-CoV-2. And I heard that this one is predominantly meant for school children and young students and graduates. So we'll try to keep it simple for everyone's understanding. And the title of my talk today is Leveraging CRISPR for SARS-CoV-2 Diagnostics. So, one of the beneficial effects of the pandemic that we have all seen, and it's also on display today as we speak, is that it has actually made science reach the homes of a lot of people, which under normal circumstances do not in normal conditions. And that is why terms such as CRISPR, some terms such as vaccines, all these things are being discussed, and people on the streets know what an RT-PCR machine is, what a diagnostic means. This is a kind of championing of science, because these things are sometimes kind of moved to regions which are considered to be too complex to understand. But due to the pandemic and due to the need to understand them better, there is more discussions happening about them like never before. So CRISPR as a tool, as a technique, is one such term which has also now become a household name, more because of the Nobel Prize that it has received in chemistry in 2020 to fascinating scientists, Emmanuel Charpentier and Jennifer Doudna, for the development of a method for gene editing. I purposefully put here that the field of CRISPR has two heroes. Of course, these are two amazing biochemists who have actually done monumental work. But like a lot of different fields, generally a field does not simply have two heroes. So therefore, one can almost edit this two out, which is what the CRISPR does, and put several heroes here, because the field itself has seen tremendous developments over almost 30-40 years to come to the point where we are talking about editing the human genome. And somebody put this up on Twitter after the Nobel Prize was announced, giving some idea about who are the people who have, over several years have contributed to this field. And as you can see, starting from the early days in the 80s till the current developments that are still happening, a lot of people who have actually contributed to different aspects of genome editing. And funnily enough, probably the most the significant contributions to this has been made by a virus or a bacteria or the archaea, which actually constitute the basis of CRISPR, which is the immune system in bacteria. Now, I told you that vaccines and immunity have become a very commonplace thing for discussion nowadays in households. But it was almost unbelievable to think that a very simple organism, of course now we can't even call them simple anymore, but a bacterial cell actually has a very sophisticated mode of immunity. And that is the basis or the blueprint for CRISPR. And this immunity is actually being developed against a virus. Therefore, the virus also has a very important role to play. So let us understand a little bit about what this immunity is all about. We have read in textbooks, we know that when we are exposed to some kind of an antigen or some kind of a foreign substance, then our body develops some sort of resistance, or it develops an arsenal of weapons, so that if there is a future attack by the same substance, then it can launch a response to that. So scientists thought that this was a very sophisticated way of handling, pathogenic microorganisms that humans have evolved into. But it was a complete surprise to them that bacteria also have a similar kind of adaptive immunity within them. And this is what is called the CRISPR pathway. As you see over here, this is a bacterial cell. A bacteria is attacked by viruses. These viruses are often called bacteriophages, and the virus tries to put its genetic material inside the bacteria, and this genetic material, most of the times, they get integrated into the bacterial genome at a very precise position, often called the CRISPR locus. And then the bacteria almost acts like a librarian. So when you have a new book that comes into the library, you try to put a tag on it so that you make sure that next time somebody wants to issue that book, you can look at the number which is present in that tag and recall that. So the bacteria is so smart that it is able to put some kind of a small tag on a very tiny piece of viral DNA, which it introduces into its own genome. So it actually introduces a foreign piece of DNA into its own genome, puts a tag on it of some kind, that's a molecular tag, so that it knows that in a second attack by the same virus, it would have a memory of that virus. Okay, so to put that into a molecular terms, when this same virus attacks the bacteria a second time, as you can see on the right side of the cartoon on the top, the bacteria uses the information of the genetic element of that old invasion of that virus, and it converts that DNA of that virus into RNA, complexes or makes an assembly of protein and RNA, which is called the CRISPR RNA complex or CRISPR RNP complex, ribonucleoprotein complex. And this entire ribonucleoprotein complex then fetches the invading bacteriophage's DNA and sits on it and chops it into pieces. Thereby, it eliminates the attack. So it used the old information of that same viral attack to ward off new attacks by that bacteriophage. So it's an extremely sophisticated and very advanced form of immunity that bacteria seem to be showing. Let us have a look at how this looks like in a normal kind of scenario through a small movie over here. What I would also introduce in the next slide is what are the main players of this whole immunity. So as you might have seen in the last slide, that there are certain terms that have been introduced over here, which is called CRISPR. Then you have heard CRRNA. Then you have also heard something called Cas, or you see something called Cas. So Cas is basically a group of proteins which, along with the RNA component, they form a complex, and then they go and bind to the bacteriophage DNA and then cuts it into pieces. So this is how this happens. So these are the bacteriophages, which actually look a little bit like alien spaceships. They are introducing their foreign DNA into the bacterial cell. It goes through the bacterial membrane, and this is the viral DNA. And since the bacteria already has an immune system against it, so it immediately brings in a small RNA piece, which there are a couple of pieces of RNA which they get together and they form a complex with this Cas protein, or a Cas9 protein to be more precise. And this complex now goes and tries to search if it has found some kind of similarity with that RNA with the new DNA of the virus. If it finds that similarity, then it cuts that DNA. So now the bacteriophage DNA is cut, so it is rendered inactive. So that's how the immunity in the bacteria actually takes place by. Now, what scientists have been smart enough is to understand that this whole complex or this whole process can actually be artificially introduced into a human cell. So once it goes into a human cell, and you preferentially tell that, this is the RNA sequence, this RNA sequence can go and bind to a same sequence on the human DNA if present. And if the human DNA contains the same sequence, then the human DNA is also cleaved or cut in the same manner as the bacteriophage DNA. Now, the thing with the human DNA is that it also has a very sophisticated mode of repair. So once the DNA is cut, it doesn't allow, the human cell doesn't allow it to remain in the cut form. It tries to break to seal these ends, and therefore the DNA breaks are kind of repaired. So oftentimes the repair is very non-specific and it makes mistakes. But in most cases, if there is a possibility of having an additional template of a correct DNA sequence, like you see over here, which has some amount of homology or some amount of similarity, then this particular piece of DNA can get introduced into that broken piece. And therefore you would have a targeted insertion of a gene. And this you can do in different types of cells. You can do this in different types of organisms, in a single cell, such as even in a germline cell, or in a non-germline cell, or even in to make transgenic animals or so. So the advantage of it is that you can actually make whatever changes that you want to make in the DNA of an organism using this technology, which is called the CRISPR-Cas9 technology. So just to summarize, this is the basis of CRISPR technology, where a double-stranded break is actually repaired, and in the process of the repair, you have this kind of introduction of a foreign DNA piece. And this is therapeutically very, very relevant, because it actually means that if there is a mutation which a person has, such as, for example, a BRCA1 mutation causing breast cancer, then one can put a correct copy of the gene in that site and then repair that, and therefore one can be able to theoretically cure that disease. So coming from that background, let us hear a little bit about how we have actually used this system in Feluda, which is a type of diagnostics which has been developed at CSIR IGIB. And this work has been done with Dr. Shobhik Maitri, is also in the panel today, and we both mine and Dr. Shobhik, we have a almost like a big joint lab where we work on elucidating different aspects of biophysics and biochemistry of CRISPR proteins. So how did we come up with the idea of COVID-19 detection? As you might know that, the type of COVID-19 tests, which Dr. Vinod had touched a little bit upon, there are three major ways by which COVID-19 or the SARS-CoV2 virus is actually detected. And they actually are answered, or basically their detection answers different types of questions. So if one does a nucleic acid test, which is also considered to be the gold standard test for active infection, then you do what is called a quantitative real-time PCR test most commonly, and the viral RNA is detected after a conversion into DNA and amplification using a kind of probe which goes and binds to the DNA. The second type of test, which often is heard about in a lot of places, is the antigen test, which basically looks for viral proteins. This is again a direct test which is looking at the infection of the virus in the current sample. Unfortunately, detection of proteins and detection of nucleic acids do not follow the same principle of amplification. Because of a technology called PCR, it is possible to amplify DNA several fold, which means that even if there is very tiny amount of viral material, genetic material present in a sample, in order to see it or detect it, one can amplify it several fold and then do the detection. Unfortunately, protein amplification doesn't happen in a straightforward manner, and therefore antigen tests have a lack of sensitivity, although it is very highly specific. Sensitivity is a measure of how often would you actually detect a viral particle if it is present even in a very tiny copies. The third test is, of course, the antibody test, which looks at the prevalence of a virus in a community, and it is not always talking about the current infection status, but rather about past infection. And Dr. Shantanu will talk more in details of how important that is for community medicine. CRISPR has entered in this field because, as I showed you in the last few slides, is that it relies on a protein going and interacting with genetic material. And therefore, the COVID-19 scenario has actually brought up a large number of CRISPR tests or CRISPR-based tests, which utilize majorly 3 different types of proteins. One is the Cas9 protein, the other is the Cas12, and the third one is the Cas13 protein. Now, the Cas12 and the Cas9 proteins actually interact with DNA, and therefore their readout is direct DNA based. The Cas13 protein interacts with RNA, so it can interact with the viral RNA, but it also goes through a first step of RT-PCR, in which it is converted into DNA, amplified, and then an in vitro transcription is done to make more RNA, and then the Cas13 based reporter cleavage is used. Among this, Feluda actually uses an alternate form of Cas9 called Francisella novicida Cas9, which we have been working extensively on in the lab in IGIB. And this principle of Feluda is that this particular Cas9 protein, which is FN-Cas9, is highly specific to point mutations in terms of discriminating between 2 substrates that are different by a single mismatch. So if you tell this protein that you need to distinguish between 2 DNA pieces which are different by a single mismatch, then there is a specific guideline in the design parameter, which if one implements, then any 2 pieces of DNA or RNA can be successfully distinguished, and the readout can be of different kinds, whether it could be a gel signature or a paper-based strip. And we had done this on different types of samples, on different types of diseases, and different types of readout. And we found that not only is it able to do such kind of disease detection very, very, very robustly, but also it can identify carriers of mutation, which is generally one of the challenging tasks through non-sequencing based approaches. Where if one of the alleles is mutated and 50% of the 50% of the sample is having the mutation and the other 50% is not, the Feluda can actually be able to discriminate that as well. And it also doesn't require any kind of PAM sequence, which is again a CRISPR terminology, won't go into details into that. So it can essentially be used for detecting any DNA or RNA mutation that is present in any genome. And an interesting study where we could actually see this being used to distinguish between 2 bacterial strains of Helicobacter pylori differing by a single mismatch, which causes rifampicin resistance or rifampicin sensitive type of peptic ulcer. And normally these kind of studies take about 3 days to confirm in a OPD, where you have to grow them in these plates and understand whether the person can be administered the antibiotic or not. Using Feluda and the RPA technology, this can actually be done in almost 30 minutes without the need for going through the culturing. So we had been doing all this for doing sickle cell anemia detection on the field before the COVID-19 actually struck, where you can see that currently the solubility test is a very crude test that the government actually generally conducts in screening camps across different villages and tertiary healthcare centers, where a blood prick is taken and the solubility is measured. It doesn't really tell very accurately about the zygosity or about the disease status. And since it takes blood, as you can see over here, there are tiny kids coming in the school to give blood, and some of them are not too thrilled about the prospect of having their finger pricked, as this tiny kid over here. So what we realize is that if we convert all this into a simple tube where the patient or the kid can be asked to simply spit into, and you have the right reagents which can lyse open the cells, the DNA comes out, and the Feluda can be done within 30 to 40 minutes, then this entire process becomes much more user-friendly. So we were doing this on a trial basis in different patients and different patient samples. when of course the COVID-19 started, and we kind of quickly adapted this to actually use Feluda for detecting SARS-CoV-2. The pipeline is shown over here. Since it's the detection is being done by CRISPR, the initial step happens like a regular PCR. In this case, the RT-PCR here stands for reverse transcription PCR, where the RNA is converted into DNA, and then this RT-PCR is done using biotin labeled primers which generate amplicons or PCR amplicons which have biotin at both sides, and then combining that with paper strip chemistry, one can have a readout where 2 bands on a strip actually makes sure that this is actually a positive patient, whereas a single band, which is the control band, tells that this is a negative sample. The entire process can be done within an hour, and because it doesn't require a quantitative real-time PCR machine which costs about 20 lakhs, but a regular PCR machine which costs something close to almost 70,000 to 80,000 rupees, one can scale up testing by almost 20-fold at the same site at the cost of a QRT PCR machine. And with more innovations, the timing as well as the number of steps have also been reduced, which we are not going into details. Again, there has been sufficient combination of CRISPR biology and paper strip chemistry. Like I mentioned, some of this has been brought in by Dr. Shobik Maiti, whose experience in nucleic acid biophysics really helped in taking this ahead. But the net result is that this is how strips look like for positive samples and a control, and negative control and negative samples is what this looks like here. We looked at the entire SARS-CoV-2 genome. Dr. Vinod very nicely gave an idea about how the sequences and the regions and the mutations in the virus makes a very important role to play, it has a very important role to play in detecting the sensitivity and specificity of the diagnostic device. So we have been doing this as well, and we are continuously doing this in trying to figure out which are the best regions in the genome which should actually be targeted, taking into account the CRISPR specific parameters, and also the parameters for making sure that we can detect up to almost a few hundred or a few tens of copies of the viral genome. And because we are both Bengalis in this team, and Feluda is of course the name of a Bengali detective. If you are aware of the Ray stories, Satyajit Ray stories, he's generally assisted by 2 assistants. One is of course his cousin called Topshe. We made a smartphone app with Aduvo based in Chennai, which can do all this quantification, and it is a machine learning app, and the final readout using just a smartphone device. And Jatayu, or the junction for the analysis of your target design of your Feluda assay, is a web server freely available where any nucleotide sequence can actually be put in, and one can get the Feluda primers and the final design of your Feluda experiment, because it is not just limited to SARS-CoV-2, but any nucleotide detection or nucleotide mismatch or SNP detection can be attempted using Feluda. Just the last slide before I finish. Of course, we have talked about the first version of Feluda, which has been licensed to Tata Sons, and then which became Tata Medical and Diagnostics. It's out in the field. The last information we received that it's being done in Delhi. But there are also versions that we have been working on making it better, where all this can be now combined in a single pot, in a single tube, which we call as the RT-RPA Feluda reaction, where it doesn't even require any kind of PCR machine, but a very small heat block. And in the most recent version, even that heat block can actually be eliminated, giving rise to a possibility of testing at a much more wider scale and closer to home, so that a return to homes, return to schools can be made possible in the coming months. And of course, the most important slide, there are a large number of people who have to be thanked for the work that has gone into this, and we are very generously funded from different agencies. So if there are any questions, I can either take them now or after the discussion.
Yeah, I think what we'll do is Dr. Shantanu will speak, and then maybe we'll take the questions. Thank you so much. It was enlightening. May I request Dr. Shantanu Sengupta ji? Yeah, we can see. Shantanu, you may have to unmute. Yeah.
Yes. Is the screen visible?
Perfect. Perfectly fine.
Yeah. So we have just heard about how genomics has been used in SARS-CoV-2, and then Jyoti gave a very nice presentation on how they have developed a diagnostics to detect feluda. Today I'll talk about Sero Survey. So what do we do in Sero Survey? So we actually measure the antibodies in the blood of an individual. So I understand that there are a lot of school children here. So in their interest, so what is an antibody? So antibodies are actually soldiers of our body. Whenever any invader, in this case it can be virus, bacteria, or any parasite, when they invade our body's immune cells immediately sends some signals, and on basis of that signals, there are proteins, glycoproteins, which are known as antibodies that are produced. So what does antibody do? Antibody, and Vinod showed this is a Y-shaped protein. It goes and binds to the antigen and then helps to process the virus and eliminate the virus from the system. So it is actually a defense mechanism, and what happens is once the antibody is there, then you're supposed to be protected. But nobody knows how long you're protected. So there are various kinds of antibodies. Actually, there are 5 different kinds of antibodies. They are designated as IgM, IgA, IgG, IgE, and IgD. So IgM is the first antibody that is produced by the body, and it is the first line of defense. So if you see in this slide, so this is where an infection, a person is infected, the symptoms starts coming, where the virus load increases. So if you can see this, the virus load increases here, and after that, after the symptoms come after a month or a week or so. So once the virus load is high, you start getting symptoms, and then during 7 or 8 days after the virus entry, the IgM, in this case, the red bar here, the IgM antibodies are generated, but they don't last for very long. They generally subside within 3 to 4 weeks. The IgA antibodies also simultaneously come. They stay for a little bit more, but then after about 10 to 14 days of the infection, IgG antibodies start coming in, and they last for a very long time. So this was known for any virus, but nobody had an idea of what was going on in case of SARS-CoV-2 virus. So when the SARS infection started, an interest on how long the virus will be stable, will there be an infection? So for example, when there is a virus load, you generally get symptoms, but it was seen that a lot of people without any symptoms also have developed antibodies, which means that there was the virus, they did not get any remarkable symptoms, but yet they were actually infected. So it is very important to understand the prevalence of these antibodies to actually assess what is the load of SARS-CoV-2 infection in our population. It's also important to understand how long will the antibodies be in our system, which will help in protection against reinfection. So based on that, we undertook a study, which is a pan-India study, where we collected blood samples from all the 38 CSIR laboratories in the country, and we looked at their antibody levels, and we basically asked 2 questions. Do you have antibodies against SARS-CoV-19? And if yes, how long will the antibody persist? So in the month of June, we started the serology. We completed the first phase in September, and now we are following up all the people who were antibody positive for every 3 months. So we are just ready with our first phase data, and what we have seen is the seropositivity pan-India is about 10.1%. Remember, there were lots of reports about herd immunity. Herd immunity is when a large section of the population is already infected and has developed antibodies. That's when we call herd immunity, so the chances of being infected becomes low. But that herd immunity is generally comes when you have almost 60 to 70% seropositivity across the country. But if you see the data from September, it's only about 10%. In some cases which are marked in green, the rates are high. So, for example, in certain pockets of Delhi, it was 25%. Certain pockets of Hyderabad, it was about 30%. But overall, the seropositivity was only 10%, which means that we are still far, far, far away from what is known as herd immunity. And we need to have vaccines to generate this kind of herd immunity. And in most of the cases, if you see these red bars, these are the places at that time. So in the month of September, for example, Trivandrum, Thiruvananthapuram, Goa, Palampur, Dehradun, Nagpur, Bhubaneswar, these had seropositivity of less than 5%, which means that we predicted that if the seropositivity of a place is low, the infections are bound to rise in near future. And that's exactly, we did an analysis. We looked at the infection rates 30 days before our Sero Survey and 30 days after our Sero Survey. If you can see here, the red circles, this is Dehradun, Nagpur, your Pilani in Rajasthan, Palampur in Himachal Pradesh, Assam, Lucknow. These all places had very low seropositivity when we started, less than 5 to 7%. And after 30 days, we saw that there is a huge increase in cases. So actually, low seropositivity is a marker for increased risk of future transmission of the virus. So this is one very important aspect of seropositivity, is that if you do a community-based seropositivity, you can probably have an indication of what is going to come in a month's time or a couple of months' time, whether the infection of a particular place is going to plateau, or whether it is going to rise, or whether it is going to fall. The seropositivity or serology experiments can tell you that. The other important thing that serology assays can tell you is how long you are protected. And what we have found, although I'm not showing here, is at least till 3 months the antibodies are present. And we have found in certain cases, even up to 6 months, the antibodies are there. So once the antibodies are generated, for 3 to 6 months, you are so-called protected, which if you have heard what Vinod said, if the strain changes, then again, you might be reinfected. And we have seen about 10-15 cases all over the world where reinfection has been reported due to a different strain. But from the same strain, once you have generated the antibody, you can be rest assured that you're protected for at least a few days till the antibodies are there in the system. And in this project, we are going to follow people every 3 months and see how long this is. The antibodies are there, which means you're protected. And this is one of the important strategies in vaccination also. If you have a vaccine, once you are vaccinated, you generate antibodies. And it is one important to understand how long will these antibodies last in your system. For example, in case of influenza, people have seen that the antibodies last for a year. So, influenza vaccines are always given every year. Similarly, there are other diseases in which antibodies probably last a little bit more longer, 2-3 years. So because SARS-CoV-2 is a new disease, we have no idea how much long the antibodies will last. So that is one of the things that Sero Survey will help. It's helping now, and especially it will help when the vaccine comes. The other way that serology will help is prioritizing people to whom vaccines should be given. For example, we are a country of 1.4 billion people, and we will not be able to give vaccine to so many people in one shot. So we need to prioritize, and you have heard in television or read in the newspaper, government has already come up with a strategy of whom they are going to give the vaccine first. So healthcare workers, people who are in the front line, and then people who interact with a lot of other people, they are going to get the vaccines. But one of the ways to further prioritize is to look at the serology. So if a person has already got an antibody, the person may not be the right candidate for giving vaccine in the first phase. So that way, Sero Survey will be very important. And this is more important because we have seen from our study that about 75% of people who have developed antibodies are actually asymptomatic. They did not have any symptoms. So prioritization based on Sero Survey probably is going to be one of the most important thing to allow us to manage the vaccine should be given in the first stage and who can be given later on. So this will aid vaccination strategy. So with that, I would like to end, and I'm here to answer any questions that you have. I would like to thank all the coordinators from all the CSR lab and all the participants of this very large effort. We have collected almost 10,000 samples from the entire country, and we are following it up every 3 months. So this is a huge effort, and it could not have been done without the help of all the laboratories, all the directors of CSR laboratories who really facilitated the study, and we are really, really thankful for them. And finally, thank you. So do we have questions now?
Hello.
Jyoti, is Kenneth sir around? Is Kenneth sir online?
Yeah, Vinod sir is online.
No, Kenneth sir.
Okay, Kenneth sir.
He is not online. Okay, you can go ahead with the questions, or otherwise I have some questions to them. I'll come in.
Vinod, you want to answer that?
Yeah, so antigens are broadly molecules which can elicit an immune response, and this could be either a humoral immune response, or this could be either a cell-mediated response. This could be a protein, this could be a small molecule. So anything that can elicit an immune response is what we call as an antigen.
Thank you very much, Juma. I think just Kenneth sir said that his connection is lost. So, okay. So we'll go ahead as scheduled, and I have a few questions for all of them. Probably the kind of condensed presentation all of them have given, we will need probably 1 hour or so to interact with them if we want to understand every possible detail of it. But grossly, since we don't have much time, and there is can you all hear me?
There is a lot of echo going on. So if someone can switch off their microphone.
Am I audible now? Is it clear now?
Yes.
Okay. So I'll just go about some questions which are not that technical in nature. When I first initiated the discussion about the program, I had a discussion with Dr. Maiti. I was interested about one thing. What could be the motivation? How did you at all think that this CRISPR can be used in diagnostics and therapeutics, and you can try this with this particular pandemic as well? What was the trigger? What is the particular trigger that you got into this line? Because sometimes I understand talking to the students that they look for such things which can make them scientists, which can make them interested in science. So if all of you and about it.
Devu, go ahead. Answer. You are part of the team.
I think
No, I think you should take this. I have talked enough. Please go ahead.
Okay, so basically we are working on developing CRISPR-Cas technology for past 2 years. So in this program, basically we are involved how to correct sickle cell anemia disease. So that is a mission mode project by Government of India. We are part of it. So during this project executions, we visited the disease affected area mainly in Chhattisgarh, around Raipur and Bilaspur. When we visited the affected area in the remote villages of this state, what we have understood, the major problem may be therapeutics, but also the diagnostics. So diagnostics, basically what for sickle cell anemia, maybe solubility, you can tell that whether the patient, whether any individual is affected or not, but you cannot distinguish whether carriers versus the disease patient and the individual distinguishing. So we thought that genotypic identification is necessary, and it is also necessary to give on site. Otherwise, once you have identified a person's disease, but you do not know whether they are carriers or trait. So after few days, if you ask them to report or to follow up, it is very difficult, because genotypic identification takes at this stage at least 7 to 10 days. So by these 10 days, clinicians loses the contact of this patient who are not anymore traceable either phone or by any means. So that was the motivation basically to start with a diagnostics, which could be a kind of POC, point of care. You do within a few minutes, few hours, and come out with the result. So you are working on that. And at the very beginning of the pandemic, when it was hitting to it already hit to China, and it was hitting to other countries like European countries, Italy, and say maybe America. So myself and Jyoti, we are having tea in our tea club, and we are discussing about this COVID-19 cases around the world. And we thought that based as it is a kind of virus and it has a genetic material, we can come out, or we can repurpose the test or diagnostics which we are developing for sickle cell anemia. So that was the trigger point. It is a small discussion between us, two scientists over a cup of tea, and the demand which we realized that test could be a very important point to taken care of, and we have a technology in our hand. So all these things actually motivated us to convert this technology for COVID-19 detections. So that is the initiation.
That is fantastic. And actually, I wanted to come to this story because I feel quite often that innovations often happens on casual discussions. When you are connected to many other similar-minded people, you get a trigger and you get into something, and then you delve deeper into it. Thank you so very much. One just practical question I'm requested to ask by one of our viewers. And that is the Feluda test available now commercially, and if not, then when is it likely to come to people?
So, I have the information as a public information which has got, circulated in newspaper. So it is available in our commercial partner is Tata Medical. Okay. So as far, news that they are basically giving test through their channel partner Apollo in 7 cities of our country. That was published, that was news and in November 9th. So I believe, it has been improved. So many more, a center has been identified. So yeah, it is available in the market, not each and every market, each and every state, because they are scaling up.
Yeah, okay. I can understand that. Maybe that's one of the reasons why, if you really look at from where we started, tests per million, and where are we now? We have come a long way. And it was a fascinating discussion in the sense that from understanding the SARS-CoV virus to actually sequencing it and using that sequence, the genome sequence, to understand what the virus is all about. To coming to the Feluda test that all of you have done, and the Sero Survey as to understand the whole geography of the country and how many people have been affected, and what the so-called the Sero Survey has revealed. In terms of the herd immunity which we are talking about, and hopefully things are taking shape and vaccine is getting developed, and hopefully I think we'll have. We are seeing the light at the end of the tunnel, and let's all hope that this pandemic comes to an end, at least by the first half of the next year. Thank you so much. And it's just one institution, under the CSIR. There are labs across the country, across different ICMR, CSIR, Ministry of Health. Everybody is working together, and including TIFR and things like that. Hopefully, this challenge which has struck us very badly, in a way, there are some spin-off benefits. One is, of course, Feluda, and there could be many more. And we are also excellent in terms of deliverables, number of vaccines which we can touch. Rotavirus is a classic case where, one of the Bharat vaccine or Serum Institute, they can come in large numbers. So thank you so much for sparing your time with our audience. I know there will be some questions in the Facebook and YouTube. We will keep answering them, and if there is any need, we will definitely get back to you and maybe seek your advice and give them the answer. I think this is the beginning of a institutional collaboration, because we are a science museum, science communicators. We connect with the school and college students. So we want the practicing scientists actually to come face to face with them to communicate this. So once again, on behalf of the Neuroscience Center, I would like to take this opportunity to thank all the 4 distinguished scientists, Dr. Souvik Maiti, Dr. Shantanu Sengupta, Dr. Vinod Skaria, and Dr. Deva Jyoti Chakraborty, and also your science outreach program, Dr. Purti of the IGIB and the CSIR lab. Thank you so much.
Sir, just before we go out, sir, hello, can you hear me, sir?
Yes.
Just before we go out, can we all come on the screen together, and can Juma take a snapshot so that we can also record it?
Juma, are you there? She's having another